Hi everyone. I'm Aniruddha Chattaraj. I'm a graduate student here at CCAM. I work with Dr. Leslow. I have been using the software for 3-4 years now. So today I'm going to show you some of the interesting features about the SpringSaLaD software and what kind of things it's capable of doing. So before I get into the details of the software, I just want to make sure that I put some documents or like some model files in that Google Drive under my name. And feel free to download this text file, this especially tutorial 1, 2, and 3, and also this PDB file here. So while I demonstrate some of the model building, we would be using these files. So it will be handy if you can, keep these 3 text files somewhere at your desktop so that when you actually build the model, you can use those. So to begin with, this is just a rough outline. So in the beginning, I'll just briefly mention about the underlying physics of SpringSaLaD, and then I'll show you how to build a model using the software. Like you can create all sorts of different things like 2-dimensional molecules, 3-dimensional molecules, and how do you define like chemical reaction or chemical rules inside the framework. And also then at last, I'll show you some of the how do you run some simulation and how to analyze the data and visualize those data. So, and this is probably one of the most important slides in this tutorial to understand when to use which particular type of simulation framework, right? So here, x-axis I have space and y-axis I have time. And you can recognize that depending on the resolution of your system, like what kind of resolution you want to look at your system, you want like different type of simulation approach. So for example, if you want to resolve your system on an atomic scale, you want to do something called molecular dynamics or MD. And there is another extreme end here, which is the cellular scale modeling, where you are interested in the micrometer size and on the time scale of like seconds, minutes, an hour, for example. Then you would do something like a PDE-based solver and stuff like that. But there is this intermediate regime here, both in space and time, where you have this intermediate resolution and you have this intermediate size of the system, where we call this mesoscale. So this is like typical size is the nanometer domain. So the size of your particle would be on the nanometer size, and also the time step of your simulation would be also on the 1 to 10 nanometer, 1 to 10 nanosecond. Okay, so SpringSaLaD falls under this particular regime here, which we call mesoscale, and that's probably it's worth remembering that particular point, that it's most appropriate for this particular region in space and time. So if you go to Google and type SpringSaLaD, that's what you get, right? I got this thing so many times, like you go to Wikipedia and type SpringSaLaD and that's what you got. But 4 or 5 years back, Dr. Paul Michalski and Leslow had some other ideas for this particular term. If you deconstruct the term, it turns out to be spring, site, and Langevin dynamics. Now, what does that mean? Let's take the first 2 terms, spring and site. So that is to say that if you have a bimolecule here, I'm showing 3 examples here. If you have a bimolecule, you can do something which in simulation field called coarse graining. So coarse graining means you are just reducing the resolution and with this appropriate resolution, you can represent this bimolecule in a coarse grained way. So if you in a SpringSaLaD type of framework, you would model this bimolecule as a collection of 2 spherical sites connected by spring. So this is the site part and this is the spring part here. Okay, so you have a bunch of spherical sites, which is to represent the interaction domain that you find inside protein molecule or in nucleic acid assembly, and you would connect those sites by a stiff spring. Okay, so that's the spring and site part. Now, what is the Langevin dynamics? Langevin dynamics is, so these are like spatial simulations. So you need to update the position of those individual particles. Now, from Newton's law, we know in order to move a particle, you need to apply force, right? So Langevin dynamics is simply the Newtonian equation with a time-dependent random force. So you see here, this is the m times a. This is the celebrated Newton's second equation of motion. And on the right-hand side, you have this force term, which is to model the viscous drag. So for example, if a protein molecule is diffusing in the solution, it will feel viscosity, right? It will feel like a backward force due to viscosity. That's what this negative signs means. And this zeta represents the viscosity coefficient of that particular medium or solution. And if there are other force present in the system, you will put it here, and there is a random force here. And the random force is to model that all the collision that you have with the solvent molecules. So for example, if there is water in the system, the protein will collide with those water molecules all the time. But you cannot model those solvent explicitly because it will be computationally a huge burden. So you model those solvent collision by by an implicit force term, which we call like a random force. And you will see how people typically model that random force here. So in this framework, in SpringSaLaD framework, so there is something called overdamp limit, where you ignore this inertial term here. So this would be 0, and you take this term to the left-hand side, and the equation becomes this. So this is the viscous term, and it is like a force balance equation here. And you have two force term here. One is coming from the springs, and one is coming from that random force. So the random force is chosen in such a way that it can reproduce the diffusion constant that you are putting for all those individual sites. So in SpringSaLaD framework, let's say if you have this molecule here, you assign a diffusion constant to individual sites, not to the whole molecule, okay? And that diffusion constant would define this is like in statistical term, this is the variance or the standard deviation, proportional to standard deviation. So how big your force can be, that would be a function of the diffusion constant, which like biophysically that makes sense, right? Because if you have like a very large diffusion constant, then the force due to that randomness can be very large because it can fluctuate a lot. But at the same time, when you have a small d value, then the force would be much more sharply picked. So your variance would be small in that case. And also the delta function is to say that the random force is uncorrelated at time. That's why it's random. So the force applied at time 1 should not be dependent on force applied at time 2. So that delta function simply ensures that uncorrelation in that stochastic force term. So that's the basic idea of the Langevin dynamics, and that's what this third part stands for, the Langevin dynamics. And just to demonstrate the idea of Langevin simulation in a little more detail, how SpringSaLaD updates the position and how it takes care of the force. So the way it works is, let's say you have this molecule in your system, like 5 sites. So these middle sites are connected to 3 sites, and the force applied to this particular site would be a vectorial summation of all the 3 forces connected coming from their neighbor, and there is a random force that I mentioned before. So this F springs that you see here, it's the vectorial summation of all the 3 forces here, and this term is the random term. So the total force applied to a particle would be summation of that random force plus that forces that are coming from those springs, right, which we call as links between those different sites. So as you can imagine, the more links that you put around a site, the less mobile it would be, because there will be high likelihood that all those forces can vectorially cancel each other, so the particle would not move a lot. So that's another way to reduce the diffusion of a site. If it's highly cross-linked with a bunch of other sites, then essentially, effectively, the diffusion constant of that particular site would be very, very low, even though you are not changing the diffusion constant of that particular site by itself. So that is to, so when like one protein molecule is cross-linked with other protein molecules in real system, you would expect a lower diffusion, right? A reduced diffusion. And that's how the software models that diffusion reduction of inside the system. And there's another point I want to point out here. This is a snapshot from the SpringSaLaD software. So one thing is all the boundaries are reflective boundaries. So we have a cubic box here. The molecules can collide into the boundary and it would bounce back because there is a reflective boundary condition that are present there. And also there are two compartments typically that are present inside the software. This yellow surface you see here, that's like a two-dimensional membrane. So you can attach a certain molecule to that membrane. So for example, if you want to model a membrane receptor, transmembrane receptor, you would attach that molecule into that yellow surface here. And you can, there is a space outside the membrane as well to mimic the extracellular events. So for example, if there is a ligand coming to bind that transmembrane receptor and it will induce some sort of conformational change in the intracellular portion, you can model that sort of process because we have three compartments inside the software. One is this two-dimensional membrane, there is this three-dimensional space outside the membrane, and there is a three-dimensional space inside the membrane, okay? Okay, so before I go into that, if you have any question, please let me know.
Yeah, I do have a question. Do you hear me?
Yes.
Yes, I was wondering, can these links or the springs change? So is it possible to have reactions between these proteins that cause binding or unbinding and so the formation of new springs?
Okay, one thing I didn't mention, I'll get into, there is something called like we can introduce bimolecular interactions. So for example, so the springs are, so for example, if this site binds with another site, right? So when they're in bound form, one more spring will be introduced here. So this site will be connected to that site through a spring. And that spring, you can introduce that spring under different circumstances. You can have all sorts of reaction conditions. I'll show you in a minute in those examples, but that will come under the reaction category. So you can define all sorts of different reactions, and those bonds that will form as a consequence of the reaction will be also treated as some sort of spring like that.
Okay, thank you.
Right, yeah.
Annie, is it possible to have the system without the membrane?
Yeah, I mean, like visually it will always exist, but you would not have any membrane bound molecule there. Yeah.
Okay.
So now I want to show you some of the capabilities of like what kind of processes that it can model. So the first and most interesting part is the second order process, which is the bimolecular binding. So individual sites in SpringSaLaD has two radius. So the user needs to define something called physical radius or rho, and the software calculates a reaction radius, which is slightly longer than the physical radius here. And as I mentioned, these are all diffusion driven process, so that will float in the three-dimensional space, right? So if they're not within their reaction radii here, if these are the two sites which are supposed to bind, if they are not within their reaction radii, they would not bind. If their reaction radii overlap, then the software will calculate a probability based on this rate constant called lambda. I'll get into that. But one more interesting thing, that the physical radii cannot encroach into one another. So there is this concept of excluded volume, right? So you have a physical radii, and that physical radii cannot overlap, but at the same time you have this reaction radii. In order to have a reaction, the reaction radii of those two sites must overlap. So that's the basic idea. That's how we model this bimolecular binding reaction. Now we have this beautiful mathematical equation here. So the idea is the users need to provide a diffusion constant, which is D, the physical size, which is rho, and the macroscopic on rate or the observed on rate, which people determine experimentally in the unit of 1 over micromolar times second. That would be our K on. So if you look at the equation, you can set the K on here. You can set the D and you can set the R as well. So R will be calculated by the software, but R is a direct function of this rho. So you can see all these different parameters. So only unknown parameters here in this equation is this R0, okay? So you have a bunch of known parameters, you have one unknown parameter, and you solve this equation and you get the R0. And this R0 is related to this lambda. So this lambda is this microscopic rate constant. So once you know the R0, you can calculate lambda. So once you have the lambda, then you can go here and calculate this probability. So the lambda times the time step of your simulation, if you multiply that, you will get a number between 0 and 1, right? And the software will draw a random number between 0 and 1. If the random number is less than that particular product, that probability, then a binding will take place. If the random number is greater than product, then the binding would not take place. So that's the stochastic part. That's like the Monte Carlo type of approach, that you draw a random number and you compare that with some sort of probability. So that makes SpringSaLaD a stochastic simulator, and it's always a stochastic simulator. And there are other processes as well which you can model. These are all like first order process. So for example, if a complex, if you define a reversible binding reaction, for example, A plus B going to AB, so the forward reaction would be the second order process that I showed you in the last slide. And the reversible process, which is unbinding process, it will be a first order process, right? And the rate constant will be always 1/2 or 1/time. And there is this next interesting reaction which we call state transition reaction. So for example, if there is a tyrosine motif in the cytosolic chain of your transmembrane protein, there might be some sort of post-translational modification where a kinase will come and make it phosphorylated, right? So you can define that sorts of state transition reaction going from U to P by this reaction called state transition reaction. So in the demo, I'll show you some of these examples, how you define those reactions inside the framework. And lastly, there is another reaction called decay reaction, which simply to say that when you have a decay reaction for a certain molecule, it will simply remove that molecule from the system. Okay, that's the decay reaction, and that's also, again, that's a first order process. And there's another interesting process which is zeroth order process, because it almost seems like you're creating something out of nothing. That's why it's zeroth order, because there's nothing on the left-hand side of your arrow. So this is a creation reaction, and that's how you inject molecules into the system. So when you have a creation reaction for certain molecules, and the rate is defined in micromolar per second. So with that rate, the software will keep injecting molecules into your system. So I just want to show you one of the interesting application of this creation and decay reaction. So for example, in virtual cell, it's called clamping. It's a very similar idea here, that let's say you want to maintain a fixed concentration for a particular molecule, right? It's like this A plus B is going to AB. So as the reaction takes place, you will deplete the concentration of A. But for example, depending on the demand of your project, you want to keep the concentration of A fixed. How do you do that? So the time evolution of A would be the following. This is the creation rate, and this is the decay rate times the concentration of A. So at steady state, this term will go to 0, because steady state means there is no evolution of system with respect to time. So as you keep this term to 0, then you get the steady state concentration of A is given by the ratio of this creation rate and decay rate. So for example, if you want to clamp the A concentration at 2 micromolar, you would define like K create as 2 and K decay as 1. And at steady state, it will give you a 2 micromolar of fixed concentration inside your system. So these are the situations where you want to use this creation and decay reaction in order to, like buffer the concentration of cytosolic concentration of some protein molecules, for example.
Could I add something to that?
Yeah,
Ani?
sure.
The reason that that's so important in SpringSaLaD is because typically we have a very tiny domain, like maybe a couple 100 nanometer cube. And that's a very small part of an entire cell. So if you have some molecule that's available in the cytosol to, say, interact with a membrane molecule in your little domain, that molecule may be very abundant, but it's going to get used up unless you have this creation decay reaction occurring to sort of feed it back in from the large world out there in the rest of the cytosol. I don't know if everybody can follow that, but it's just a way to make sure that there's enough of this A molecule present to really represent how much is available from the cytosol if A is being bound to a small number binding sites at the membrane. You never want it to be used up because there's always more that can come in.
Yeah, right. So yeah, so next time I'm going to show you some of the demonstration of some biological system, but in case if you have any questions so far, feel free to ask.
I have a question regarding what Dr. Lowell mentioned, that we usually know that in biology, the diffusion is in the cytoplasm. For example, it's in an order of 10 micrometers square per second or so. So it means that it's not just a nano domain. So how reasonable that we can answer some biological question in a nano domain? That is the question by using the model that you just mentioned.
So it does some internal conversion to convert. So in the interface, you enter the D value in terms of like micrometer square per second, but the D value that it actually used to like solve all the numerical equation, it's on the order of like nanometer square. So there is this internal conversion there. And I mean, in biological system also, if you think about the actual real system, the size of like individual protein domains, they also fall into this nanometer domain, right? They're not like micrometer domain, like the size of individual protein molecule. So it all falls into place, I guess. It's just that there is this internal conversion between this micrometer square to the nanometer square, and it works, like it does all those things internally to keep it consistent. Okay, so I'll just move forward because I wanted to show some of the demonstration stuff here. So the system that I'll be showing you today is this ARP2-3 dependent actin branch formation. So the main idea, you might familiar with this system, that actin dynamics is a very important process inside living cell, because it does like a whole wide range of function inside cell. And there is this interesting process that is actin branch formation. So in order for that step to happen, you need existing F-actin or actin filament. And you also need a complex called ARP2-3 complex. And this ARP2-3 complex is not active always. So you need another factor called nucleation promoting factor, or NPF. So when ARP2-3 complex binds to NPF, it gets turned on. And then it binds to this existing F-actin, and then it can recruit like a monomeric actin from the solution. And this particular side branch will grow, okay? So we will take like a one particular portion of that pathway, and we'll try to simulate that using SpringSaLaD. So this NWASP or WASP, they belong to that category of NPF, or nucleation promoting factor. So here you have this, at the extreme C terminal, you have this domain called VCA for WASP, and you have 2V for NWASP, and you have this followed by this domain called proline reach, polyproline residue or proline reach domain here. So the interesting fact about this complex is that ARP2-3 complex can bind to VCA at the same time. So it has two binding sites for VCA, and experimentally it has been found that when ARP2-3 is bound to two VCA simultaneously, then it's in the that's the most active form of ARP2-3 complex. Okay, so in our SpringSaLaD representation, that gets translated to this. As I mentioned before, in SpringSaLaD, we always model a biomolecule as a collection of spherical sites. So here, for the sake of resolution, I'm showing you as 3 different or 4 different blocks, but in my model, I would model this whole thing as a single sphere, okay? So there's this another idea here, that there's this something called allosteric activation. It's very abundant in all biochemical system, and most of the biochemical system, that the ARP2 first binds to one VCA, and that VCA binding to that particular domain induce a conformational change which alters the affinity of these sites. Okay, so the VCA binding of this light green will alter the affinity of this cyan to this second VCA. So there is this allosteric activation going on here, and we will see how can we model such process in SpringSaLaD. Excuse me. And in the green sites, so just I didn't mention that, so these green sites are proline rich motif, which has, so it experimentally has been found that in that particular region, we have this 6 proline rich motif, which is this particular stretch here, which is a disordered region in the protein structure. And when it can interact with this SH3 domain containing protein. So here I'm showing a protein called NIC. So NIC has 1 SH2 domain and 3 SH3 domain, and those individual SH3 domains can interact with the proline rich motif. So I'm showing here 2 different binding modes because it's all these are all possibilities, because in one case all the 3 SH3 can bind all the 3 PRMs of the same NOS molecule. But in another case, one NIC molecule can cross-link between 2 different NOS molecules, right? This SH2 domain can bind to this transmembrane receptor protein, which is the phosphotyrosine here. So the SH2 is a traditional signaling binding domain for this phosphotyrosine. And you can imagine, due to these multiple binding sites, it can all cross-link within themselves and form this signaling cluster. So it's also one of the very interesting research topic, that how all these different molecules cluster together and what kind of role it has in the downstream signaling process. So I'll try to show these 2 different processes using SpringSaLaD and how we can gain some interesting insights from that type of modeling approach. So this would be my first demo. So here I have a NIC molecule with 1 SH2 domain and 3 SH3 domain. And here I have my NOS molecule with 6 proline rich motif. And these interactions are it has very weak affinity. So SH3 will bind to the PRM with an affinity of 100 micromolar. And another interesting thing, there is SpringSaLaD uses something called rule-based modeling, which you would hear more from Dr. Michael Blinup tomorrow. That is to say that there are 18 binding possibilities here, right? 3 SH3 and 6 PRM, so 18 interaction. But in the software, you just need to define this one single rule, and it will take care of all the possible 18 interaction. So that is pretty convenient when you're dealing with this multivalent system. And in a lot of cell signaling system, you see this system more often. And there is this another component here, which is this tyrosine, this nephrine molecule, which has these 3 tyrosine residues in his cytoplasmic tail. And that SH2, which is the yellow site here, it can interact with each of those red sites with affinity of 1 micromolar. So that's our interaction domain, that's our interaction module. And now let's build the model. So, so I guess you should have this jar file already installed in your desktop. So the way you build a model, that you just launch the jar file. And once you get into that, you come under this molecule tab and you say add new, and you rename the molecule. So we will say this is NIC. So remember, NIC has 2 sites. One is SH2. I know the radius of the size. So this sizes of this radius of the sites would depend on the kind of system you're dealing with. So you want to put that kind of size for your particular system. So for me, it's like SH2. It has a typical radius of 2 nanometer. So I'll just do that. And then I'll add 1 more site type called SH3, and the typical radius of SH3 is 1.25, and I'll give it the color magenta to be consistent with my other cartoons. So, and then once you define the types, then you add site here to build the molecules. Okay. So just for the convenience, I'll keep it apart here. And so I just added 1 SH2 molecule here, and I'll add 3 more SH3 into my system. So just you can define the position of those individual sites in this graphical editor. So I'll add 3 sites. Second SH3 and third SH3, and you can set the position here. So now I have defined one SH2 and 3 SH3 domain, and that completes my Nick molecule. It won't be complete until you define the connectivity. So remember all the springs that I was talking about? So here you define the springs or those links. So I'll put 3 links sequentially between all those different sites. So site 3 to site 2. So now I have 3 links, site 0 to site 1, site 1 to site 2, and site 2 to site 3. Okay, so that will be our first molecule. And at this point, it's a good idea to save the molecule. So you can just save the molecule here. I'll call demo 201. And it will give a pop-up saying, do you want to create a project folder? You can press yes, but I'm used to pressing no, so I'll just go ahead and press no, because the directory structure will be a little bit different, but it won't change anything else in your simulation. So I have one molecule, then I'll build one more molecule called NWASP, and NWASP will have site type called Proline reach motif, and the diffusion is fine. And yeah, I mean, you can put any color you want. It's not a big deal. But I was just looking for the green color. Now I'll just put 6 of them, right? So because there are 6 Proline reach motif here, so I'll just put 6 of them. Okay, I mean, ideally I should put 6 of them, but for now I'll just put 4 because you get the point, right? How do you do that? Just for the sake of interest of time, I just put 4 sites for now, and that's how you build the model. So for example, then you just create the link like site 0 and site 1 and site 3 and site 2, and that would make your model complete. So, so once you've created your 2 models, then you can go here and define your binding reaction. So for this model, we'll just have 1 binding reaction, and this is the idea of rule-based approach. So you'll go to the Nick molecule, go to the SH2. We'll just define the state for now. We'll get into that state thing, how to define different states and how to use that. So for now, it's the reaction is saying that the SH2 of Nick molecule will bind to the PRM of NWASP, and all the possible combination will be taken care of. So we'll call it SH3 PRM binding, and you will give on rate and off rate. So typically, if the kinetic parameters are known, you are expected to put those numbers here for the on rate and for the off rate. But most of the time, you just know the affinity of the interaction. So the biochemical affinity is essentially the ratio of the off rate over the on rate, right?
Ani,
So,
interruption quick.
Yeah.
I'm not sure where you're going with this, but usually it's the SH3 that binds to the PRM.
oh, I'm sorry. Yeah, okay. Yeah, I'm sorry about that. My bad. Yeah, right. So it will be the SH3 of Nick which binds to the PRM. Yeah, I'm sorry. So the rule is saying the SH3 of Nick would bind to the PRM of NWASP, right? So now we have 2 molecules and 1 reaction rule. We'll save this model, and then we will launch a simulation to see what's going on inside the system. So the way, so once you press that tool simulation manager, you will get this window and you will see all these buttons here. So just to add a simulation. So when you add a simulation, you'll get this pop-up. It says couldn't find the PDB source file, just ignore it for now. It's we will get into that. So this is the simulation 0, and here you can change some of the simulation parameter here. So for example, right now we don't have any Nick and NWASP to begin with, so we'll just put 5 of them each. So it's so the simulation box we have, you can change the size of the simulation box here, like X, Y, and Z direction. And these are the initial conditions. So we just want to see something quick that what's going on. So, and this is the one disclaimer that it might take a really, really long time, like maybe days or something, if you are trying to simulate a very big system. So that's why we will always, for the sake of the time, we will always make a very small system and show you how to run the simulation and visualize the data. And then we have enough time, you can play with the concentration and numbers of those, your model components. So you saw our simulation is finished now. So this is the time you can visualize the data or see the data, for example. So there are two tabs here. One is called view data and one is called view 3D snapshots. When you click at the view data, you will get this nice table here where you can find a lot of different type of data. So the data class section says what kind of data you want to see. So this is the molecular counts. So molecular counts means Nick, total Nick will be always 5 because there is a mass conservation here. And how about Phrenix? So I start with the Phrenix 5 molecule, but as time goes on, it decreases, right? And you will see the bound Nick is increasing. So this is the bound Nick, and this is the Phrenix. So you can get the information from like how many molecules are in the bound state, how many molecules are in the free state. And you can have the bond data as well. So initially we didn't have any bond, but then as time goes on, it forms a lot of bonds inside the system. So you can get that information here. And one of the particular interesting behavior or interesting data for this kind of system is something called cluster size. Okay, I did something, it didn't work, so I forgot to mention that. So when you come into the data construct section, there is something called cluster counts, which are unchecked by default. So you want to check that button here. All the other properties are checked by default. So remember that if you are dealing with this type of multivalent clustering, you come here and check that button here. Now, if I go and launch another simulation, you can see that clustering behavior. Oh, I'm sorry, I forgot to add molecules. I can just rerun this one. So you just override all those data and you can rerun the simulation one more time with your updated data constructs and updated simulation feature. So it will take a few seconds to complete, and depending on the kind of machine you're using, it might take like a few seconds to a few minutes if you're dealing with fairly small system. And now you can view data, and in the view data you have this cluster data tab. Oh, okay, that's weird. I think I didn't update it properly. Let me just run one more time. Sorry about the confusion. So there is another feature I just wanted to mention, that you can run things in parallel. So for example, you want to run 4 trajectories, because these are all stochastic trajectories. In order to get a meaningful result, you want to run multiple trajectories, right? So let's say we want to run 4 trajectories, and you can say, okay, I want to run them in parallel. So you check the button and it will ask you how many runs you want in parallel. So I'll say 4, and if I launch this simulation again, and as usual, I forgot to put the initial condition, and if I put initial condition here, and then I run one more time, now you can see that all 4 of them will be running simultaneously. So at this point, my CPU is making a lot of sound because these are all computationally intensive stuff. So, but you can that's the feature that are available that you can avail within this particular framework. So now going back to our simulation, I want to show you. I hope I have the cluster data this time.
I'm going to interrupt again. Sorry, just to make sure people realize that you can send these simulations to a cluster.
Yeah, like right. If you have like access to high performance computing facility, then that's what I use, and that would be the ideal thing to use, because your local machine might not be compatible to handle this larger system. So if you use like some sort of computer clusters, that would be the ideal thing to do, just using some Linux script, and you can send multiple jobs. And once you, SpringSaLaD has a way to collect all the data, but for the interest of time, I'm not going into that, but it's all available in that tutorial that we have in the website. So once you have like multiple trajectories, SpringSaLaD can do all sorts of analysis for you, combining all those trajectories and give you the average behavior of the system. So, so that's it for this particular section. I'll just go ahead and show you some of the results. As I mentioned, that it's a little tricky because it takes a lot of time. So I'll just show you some of the results that I already got by using some sort of larger model. So for example, you remember we have this 3-component system, nephrin, NIC, and NWASP. It's not a trivial thing to imagine that what would happen if you increase the biochemical affinity of the system. So I ran this virtual experiment by using 2 different affinities between all those sites. So the blue one, you can imagine the affinity I was using for the blue one was that I just showed you. So 100 micromolar for SH3-PRM and 1 micromolar for tyrosine and SH2. And once I increase the affinity by a factor of 2, I get this increased cluster size into the system. So you get bigger cluster into the system once you increase the affinity. But that might not be the interesting part. The interesting part is the distribution. So once it reaches the steady state, now you can observe something interesting here. For the blue one, for the lower affinity, you have like a little wider distribution in that cluster size. But for the red one, so once is the mean of the distribution is shifting towards the right, but another interesting behavior is that the clusters are more discrete in nature, right? For example, this cluster size 11 is the most predominant one here. Now, that's a very interesting observation. Now, if we try to understand why it's happening, we need to look at the binding stoichiometry of all those multivalent sites. So remember, nephrin has 3 NIC binding domains. So the binding ratio of nephrin to NIC would be 1:3. And NIC can bind half of NWASP because NIC has 3 binding sites, WASP has 6 binding sites. So the stoichiometry of NIC and WASP would be 3 to 1.5. That if you sum that up, you get 5.5. And 5.5 is a fraction, and we are dealing with number of molecules. So number of molecules cannot be fraction. So if you multiply the number by 2, you get that magical 11 here. And you see that that 11 is the most predominant one. So the underlying insight from this simulation is as you increase the affinity, that binding stoichiometry is driving that cluster formation. So if you have a very tight affinity, so only clusters with this favored only clusters with this perfect stoichiometry will be favored. But if you have a lower affinity, then you can get like a wider distribution of cluster. Okay, so that's the biochemical. That's the kind of insights you get from this type of simulation. So for the interest of time, I'm not stopping for question at this point. I guess we'll get back to the question at the end of my demo. I want to show you other 2 like cool features inside the software. So then we can discuss all the results and if you have any additional questions. So there's this another feature in SpringSaLaD that you can build the molecule automatically. So for example, this is the NIC actual structure of NIC molecule, 1 SH2 and 3 SH3 domains. And so the thing is, only the structured domains are experimentally resolved. You cannot really solve this disorganized domain here, which is the linker sequence here. So in order to get this and generate the PDB for the whole molecule, I use this structure prediction software called Raptor X. So what it does that it does some sort of sequence alignment to correctly, and it generates some sort of rough structure for this disorganized region here. So the PDB that I put in the Google Drive is exactly this one. And in SpringSaLaD, there is a way to build a model component directly from this PDB structure. So we use something called k-means clustering algorithm. So k-means, what it does that it will ask you that how many sites or how many domains you want for this particular molecule. You would say, I want 4 of them. So then it will generate the structure for you, okay? So the color coding you need to do by yourself, but it will generate a structure for you. So if I can show you that feature very quickly. So the idea would be, let's say we are this particular window and we want to add that I'm sorry, you go to here, add PDB from file, and I have this nic1 human.pdb file here, and it asks me how many sites I want. I would say 4. And if you want the coordinate, if you want to give the coordinates of individual sites, that's okay too. But for most of the cases, we don't know that, so I'll just press cancel, and you will see that molecule will pop up here. So here you can see that behavior. Okay, so this would be our SS2 domain, which is slightly bigger than the SS3, and these are the 3 SS3 domains. So you can also see it can also generate the proper geometrical arrangement for that structure as well. So this is just one of the advanced feature, but I wanted to show you that that feature is available. You can build model directly from the PDB structure if the experimental structure is known for that particular molecule. Okay, then next thing I want to show is this allosteric activation that I mentioned. So we have this representation of ARP23. So ARP23 has 2 sites, right, for VCA, like the site 1 and site 2. So the idea is that site 1 is 0.1 micromolar, and the native affinity for the site 2 is 1.5 micromolar. But once this site 1 is bound to VCA, then it will switch to a high affinity state. Then it can bind to the VCA with an affinity of 0.1 micromolar. Now, how do we define this reaction or rules inside inside SpringSaLaD? So I won't build the model from the scratch. I'll just show you how to define those reactions. So this would be our model representation. We have ARP23, like a big structural domains. Like it's known that you have like 7 subunits for this ARP23, and you have 2 binding sites. I'm calling here ARP2 and ARP3. So one of the interesting feature is that you can add states to those individual sites, right? So I have defined 2 states here for ARP23. I'm calling it without VCA and with VCA. That is to give a proxy that whether this site is bound to VCA or not, right? So depending on that, it can remain in these 2 different states. And depending on that, ARP23 can be in these 2 states, this low affinity state and this high affinity state. So once you define that molecule and with those different states, you go here in the binding reaction tab, and you define that the VCA will interact with the ARP23, and the state is without the VCA, and the affinity is 10 and 1. So the affinity would be like 1/10, which is 0.1 micromolar. And that binding will induce this transition reaction here. So the ARP23 will go from without the VCA to with VCA state, right? So the condition is when ARP2 is bound to this VCA, that's the condition, bound. And which molecule is that? The conditional site is VCA, conditional state is state 0, because we don't have any state for VCA, so state 0 is fine. That state transition reaction will take place. And there is this reverse process as well, that when it's free, then the reverse process will happen, like with VCA to without VCA, right? So once that state transition takes place, then you define another reaction. So remember, the previous one was without VCA, and this one is with VCA. And this is something that you need to define in order to help the software to function properly. Because if you don't define the second reaction, it would not know what to do with this particular state, because you will have a state transition reaction here, but it can no longer find this particular string, so it will throw an error. So whenever you have a transition reaction, you need to have 2 different versions of the same binding reaction, like this. Okay, that's well explained in the tutorial as well. You can go ahead and check the tutorial. And another interesting fact about that particular allosteric process, that now we will define allosteric reaction called ARP23 is going from low affinity to high affinity, and the condition is when ARP23 is in this particular state. So when ARP23 in this particular state with VCA, then ARP3 will go from low affinity to high affinity. So that's the allosteric transition that we're talking about. And that's the reverse process, that when it's without VCA, that means ARP2 in free form, then it will again switch off, like go from high affinity to low affinity state. So that's how you model allosteric reaction and transition reaction inside the framework. And for the interest of time, I would just show you some of the results that I got from using these two models. So this was the simple version of model 2 and the earlier was model 1. And I combined these two models. So here I have 2 NWASP with 1 VCA, which can bind to ARP23. And in the upstream portion, we have this NIC, which can recruit this whole machinery to this membrane-bound receptor here, which is the nephrine molecule, right? So what happened when I have just 2 NWASP and 1 ARP23? So in this plot, what I'm showing you here, that what is the fraction of bound ARP2 and ARP3 at the steady state, and the blue one corresponds to the state when both the sites are in bound form. So remember I mentioned this full bound ARP23 is the most potent form of the actin nucleator. So this would be your reference system, that when you don't have anything else, just 2 NWASP and 1 ARP23, 65% of the time ARP23 remains in this full bound form. But look what happens when I include that nephrine and NIC molecule in the system. So when you include the nephrine and NIC molecule in the system, that full bound fraction shoots up to 93%. So what is suggesting that when you recruit all this machinery near the membrane, due to this effective high local concentration, you are essentially promoting the actin nucleation potential of the ARP23. So these are the different kind of things you can explore using SpringSaLaD, and it's really helpful because it allows you to play with this effect of this local confinement, and since it also allows you to model the structure of the molecules, you can also probe the effect of structural rigidity or structural flexibility and the role of steric effect and all sorts of different stuff. Like, for the interest of time, I couldn't show you all the different things, but I would encourage you to check our recent work that we published last year. I put that link of the paper in the Google Drive as well, where we actually looked into more detail that how does this different structural parameters can affect this clustering behavior, and that clustering behavior might also have significant influence in this downstream process as well. So yeah, I think I'm done. I just want to take the opportunity to thank all my mentors and all the people associated with the SpringSaLaD project, and also like CCAM to give me this opportunity. And now it would be a good time if you have any question.
Hi.
Hi.
I'm Leo. Quick question. What kind of, how can you output different types of data files, or how do you output the actual data?
Yeah, I forgot to show you that. Okay. So that would be like a typical data structure here. So for example, I just run one simulation right here, and so this is the typical file format that you have. This is your simulation file, and there will be a directory called sim folder. And in that section, you have all the different data files in the CSV format, for example. So you can clearly use this file, and like these are the files you can open with Excel and plot all sorts of different things. So this is run 0. You have all the data like full bond data, full count data. These are the site data and all sorts of different data file. You can look those data here. And there's another file called viewer file. So it gives the coordinates of all those sites as a function of time. So here you have all the coordinates of all the sites and their connectivity between those sites. So you can also process these files and do your own analysis if you want. So this is all very useful. It generates a whole bunch of raw data. It needs some post-processing at this point. So we are also working on that to provide some of the functionality. But yeah, so these are the location where you would find all of your data.
I also have a question. I was wondering for the SpringSaLaD, so it's obviously for a limited amount of molecules. Like how many are typically calculated? Like how far can you go so that it's still feasible?
So I mean, that's a very tricky question. So for example, like this example I showed you here, I had so So this was our model, right? Like Nick, wasp, and nephrine. So in here in that particular plot, I had 10 nephrine, 30 Nick, and 15 NWASP. And in order to, I ran those individual trajectory for 800 milliseconds, and one trajectory took around 20 hours. And that would also depend on what kind of machine you're using. If you're using any computer cluster, that might vary a lot. But typically, you want to remain on the order that when the number of sites are on the order of 100, like 100 or 200 maximum. Because if you push too much, then it just you might need to wait for like 2-3 days or something.
Okay. Yeah. Thank you.
Any more question?